Good morning, all of you. Today we are going to discuss about the preparation of the nutrient agar, slant and star culture. So all of you know why we are doing this practical. The previous class, so what we have done in that it is the same thing only, slightly modified. There we perform the practical in the petri dish. Here we are going to perform the same practical in the test tube in the different principle. So what is the use of the slant and star culture? First we are going to see the game or what we are going to do and compared to the petri dish what difference it is. First, as usual like the petri dish, here also we are going to prepare the bacterial culture. Second, refrigerated and subculture. Here it differs when compared to the petri dish part. That also comes under the category of the isolation of the microorganism and isolation of the pure culture. These also comes the same category, but the principle varies. Preservation and subculture. Usually in petri dish, so we do the mother culture, we bought the mother culture. Do you know the difference between the mother culture and subculture? What is mother culture? Any of you have idea about the mother culture? So mother culture is nothing but the original culture developed from the laboratory under the particular microbial species. For example, in E. coli, particular E. coli, if you want to preserve in a mother culture, means first we will buy from the industry side, then we took to our lab, and here we will do the subculture. Subculture is nothing but taking one loop full of the mother culture, and daily we are doing subculture for our practicals. Why we need to preserve the mother culture and why we have to do the subculture? What is the importance of the subculture and what is the importance of the mother culture? Mother culture shows the complete viability. It shows complete viability for a long term. So if we spoil or if we contaminate the mother culture, we won't get a good species for our practical. That is the reason why we won't disturb. We take a first loopful of the culture, thereby we keep on doing subculture. If the subculture is contaminated, nothing will happen. There is no need to buy. Again we have a mother culture and we can take the same thing. But once if you contaminated the mother culture, the entire thing completely waste. Again we have to buy the same mother culture from the same place. So to avoid such a things, we prefer especially subculturing for practicals. Next, what is the principle behind the nutrient slant and star culture? So slant and star both we perform in the test tube only. So here the methodology varies for the slant and star. Star, you know, stabbing means we have to deeply insert the device to something, medium or body, whatever object, whatever. Stabbing is nothing but deeply inserted the device. Slant means we give a slanting position. So here see, this is the star culture. Here we are going to inoculate a microorganism 1 centimeter depth. After preparing the star culture, okay, 1 centimeter depth with the help of the inoculating loop, we deeply inserted the culture inside the medium. But slant, the same like petri dish, where exactly the slope starts and where the slope ends. Starting slope and the ending slope. That is the streaking position. Here we perform the simple zigzag streak. That is the principle. So preparation, everything is same. Working formula, everything is same like the petri dish. Isolation of the nutrient, what we have done the same working formula. Here also we are performed. Next, so what is slant? Bacteria born in a sloped surface. That is a slant position. The slant surface, bacteria achieve their growth. So what is the use of the slant culture? Here, as per the use, both they varies. What is the use of the slant culture? Slant culture is used for the routine subculturing and maintenance of the subculture. Here there is no mother culture where anything is there. No. So the use of the slant is nothing but doing routine subculturing for the lab practicals. We perform slant by taking a mother culture. Next, stab. The same thing. Okay, we deeply inserted the culture in 1 centimeter depth inside the test tube. What is the use of the stab? Why we deeply inserted the culture inside the agar medium in the deep level? Why we do the strip on the surface of the agar medium in the slant? What is the difference? The mechanism is totally different based on the classification of the anaerobic and aerobic. Okay, so the slant, the aerobic microbes, they achieve the growth because they contact with surface oxygen. So when we deeply inserted the inoculating loop along with the culture inside the agar medium, then there will be no oxygen is there inside the deep agar. In the surface, oxygen will be there. Now, if you agree this point, over the surface like the petri dish, test tube also, oxygen supply will be there. Inside the agar medium, will be oxygen? No, not at all possible. So, if we perform the same practical under the stab culture, if the microbes comes under the category of the anaerobes, then they will survive. If the microbes doesn't come the category of the anaerobes, they won't survive or they won't achieve the growth. That is the main reason we are doing slant and stab. So, what is the use of stab? What is the use of the slant and stab? Why we perform the stab culture? Why we perform the slant culture? Slant culture means immediately in your mind strike that it's for aerobic bacteria. Stab means it's for anaerobic bacteria. Next, the main use of the stab is nothing but long-term preserve and motility. So, if you want to preserve the culture for the long term, so we need to do the, or we prefer the stab over the slant. Okay. Next, motility. For motility also we need the long-term preserved viable cultures. Okay. So that is the reason why for slant, routine subculturing and maintenance we prefer. If you want to cultivate the anaerobes, if you want to cultivate, if you want to grow the anaerobic microorganism for the long term, as well as if you want to do the practical for the motility, then the preference goes to the stab culture. So this is the today's discussion, and you are going to do the practical like the same manner. Okay. So same hot air oven. Next we will go for the autoclaving, then laminar air flow. So you make your group according to the instrument what you are going to use. First group, go and measure, cleanly measure it, and autoclaving group. So be cautious and careful, because of this reason only they did the mistake. Okay. So do you have any discussion about this class? Any questions coming from your side? Any clarifications? Okay.